right. Let's see. Good time of the day to everyone. My name is Alex Kopyov. I run research and development for a small stem cell startup in California. We work to develop treatments for neurodegenerative diseases, and we're somewhat unusual in that we use undifferentiated cells as a primary substrate. We think they work better than most of the research directions tried today, and I will try to show you why on the example of Parkinson's disease and several others. When professionals think of Parkinson's disease, dopamine deficiency is something that comes to mind immediately. However, what is not thought often is that's not the only thing that's happening. Parkinson's disease is a multi-factorial disease. Besides dopamine deficiency, you have serotonin deficiency, which also contributes to motor impairment, but it also causes cognitive decline and mood changes. There is also acetylcholine surplus, which causes dementia. There's also glutamine surplus, which causes neurodegeneration. And if you think about it, ultimately, it's not dopamine deficiency or motor degeneration that kills most Parkinsonian patients, it's dementia. And somehow most treatments ignore things other than dopamine deficiency. The current treatments of choice for Parkinson's are not that many if you break them in classes. L-DOPA, which is basically a dopamine deficiency, has been around for ages. And it has a lot of problems. It causes tolerance. It doesn't really arrest the disease progression, so it's not curative, it's just symptom control and motor symptom control only at that. And the worst thing that once the tolerance is established, the side effects from L-DOPA can often be worse than the symptoms of Parkinsonism, are plain intolerable for the patient. Deep brain stimulation has been pretty much a godsend for Parkinsonian patients because it can give a very good control of motor symptoms alone. And that lasts for a long time. However, it still does not stop the disease, and these patients eventually die when they develop dementia. Moreover, it requires constant fiddling with the settings. They say once you get deep brain stimulation, you're pretty much married to your neurologist because you have to visit them constantly. So that is a, very good option. However, it's still not where we want to be with Parkinson's. There's also physical therapy, but again, it's helpful, but it's only about symptom control. What is being studied today? As far as regenerative medicine, there is induced pluripotent cells that are usually pre-differentiated in dopaminergic neurons. There is various adult pre-differentiated cells, again, pre-differentiated into dopaminergic neurons. There is still fetal tissue transplantation, which works through replacement of dopaminergic neurons. One thing about these three is that all of them, again, work on dopamine only. And all of them have a huge side effect, which is, for example, why fetal tissue that has been tried the first time almost 30 years ago is not really mainstream yet. It's that you cannot dose them. Unlike L-DOPA, that if you give a patient too much L-DOPA, you can cut back. Once you implant dopamine-producing cells, if you implant too many, this patient has to live with these horrible side effects. There is no way to reverse that. And that has been a major stopping factor for trying all of these three. So it's frankly puzzling to us why people still continue working in this direction. There's also gene therapy. However, it's very new. Very, very small percentage of Parkinsonian patients are actually, their Parkinsonism is of genetic nature. And we know so little about the genetics of it that at this point it's just scary to get there, because you can affect one gene and cause such a cascade of other changes that at this point I think it's way too raw. There's also antibody-based treatments, but similar problem as with gene therapy. We don't really know still what causes Parkinson's disease. We have some theories, but nobody knows, and it's probably more than one thing, but pathogenesis is still not understood. So antibody-based treatment at best address something that accompanies Parkinson's disease. We don't know if it causes it. We don't know if it's caused by Parkinsonism. And the latest antibody-based trial where they had antibodies against alpha-synuclein has failed. There's still pharmaceuticals being developed, but again, they're mostly geared towards dopamine replacement only. Oops. So why we think undifferentiated cells are a better option for any of those? Because an undifferentiated cell, just like it happens in an embryo, can respond to multiple environmental cues. So when a dopaminergic neuron dies, that necrosis releases multitude of factors that stem cells can react to. They can multiply and differentiate into according adult cell type, in this case, a dopaminergic neuron. If multiple cells are involved, as would be with Parkinson's, they can, they can develop into glia as well, or other cell types. One thing that we found out that this kind of multiplication and engraftment is not the only thing is going on. These cells tend to release neurotrophic factors, the factors that are known to provide neuroprotection, factors that stimulate brain's own regeneration, and harness brain's own resources, and produces very good effect. Another consideration is that you can use undifferentiated cells without modification to treat multiple different kind of diseases, because they're undifferentiated, they're fairly universal. One thing I haven't mentioned is that the cells that we use are fetal derived. So there's a fair amount of stigma associated with that, but we think it should be overcome, because they tend to have the best of both worlds, where they're non-tumorigenic, unlike embryonic cells, but they also have their potential for developing into multiple cell types. But the biggest, I think, advantage of undifferentiated cells compared to, say, induced pluripotent cells is that undifferentiated cells work on a negative feedback. They can stop when there is enough. So we do not really to concern ourselves too much with dosing. When they start producing enough dopamine, they stop. There's no, there's no side effects. One of the big challenges with working with undifferentiated cells is that most existing animal models, if you look at preclinical scene, do not really work. The most common models that have been used forever and ever in Parkinson's disease research are neurotoxic models, such as 6-OHDA and MPTP, which selectively kill dopaminergic neurons in the brain. Apart from this is, again, a single factor simulation. This does not simulate Parkinsonism, it simulates merely dopamine deficiency. But the biggest problem for specifically undifferentiated cells is that this is a static model. Once you inject 6-OHDA and it kills dopaminergic neurons, nothing else is going on. You get scar where substantia nigra used to be, and that's it. So when you when you implant cells at that point, the cells have no microenvironmental cues to respond. This is in opposition to what's going on in human Parkinson's, where you have a continuous neurodegeneration. You have a continuous neuron death. In that case, implanted undifferentiated cells have something to do. They know where to migrate, and they know they need to replace dopaminergic neurons. There is quite a bit of work also going on with transgenic models. Unfortunately, that is still raw. They have inconsistent phenotypes from batch to batch, from litter to litter, and within the same animal, as we found out. Pink One, for example, a model sponsored by Michael J. Fox Foundation, now experience symptoms come and go on their own, which makes it extremely difficult to do any kind of efficacy studies. This is why we've been working a lot with the spastic hand-Wistar model of this is a little-known model. There are only 2 colonies of these kind of rats in the world that I know of, but we were privileged enough to work with California State University Northridge, which has one of those colonies. It's actually a model of ataxia more than Parkinsonism. It's characterized by spontaneous mutations, which are inherited. But the biggest thing that we like about it is that it's a model of progressive neurodegeneration. Purkinje cells in cerebellum slowly start, well, not slowly, actually, they start dying off rapidly, but it's over a period of time, and the cells, undifferentiated cells, have time to react. It's really, really good model for studying because of its rapid course. These rats develop motor symptoms at age of 30 days, and by the time they're day 60, they need to be put down because the disease progressed so much they are not able to move. And the symptoms are severe, and they're easily quantifiable. They're consistent, and so you know what you start with, and you know whether your treatment worked or not. And this is what happens when you implant undifferentiated cells into a cerebellum of such rat. This is a needle track on the left, and you can see brown dots are the stem cells. This is human cells implanted into the immunosuppressed rat. And you see kind of like a mad rush of these cells moving towards the Purkinje layer. These huge, big things are Purkinje cells to repopulate it. So again, being undifferentiated cells, because there is a neurodegeneration going on in Purkinje layer, they feel it, they know it, and they move in to fix it. Clinically, this is what you see accompanying it. This is an open field test, which tests the distance traveled by these rats. So the higher, the better. An untreated normal rat is represented by a blue line. This is what it looks like with a normal rat. A yellow line is a spastic rat, a rat that developed ataxia, and you can see deteriorating here. And then it got treated with stem cell injection to a cerebellum, and it rapidly recovers and then is pretty much indistinguishable from a normal rat. If you look at the controls, a gray line is a dead cell control. We kill the stem cells implanted into the brain, and you can see that it deteriorates rapidly and it has to be put down at day 60. So it's quite stark contrast compared to treated animals. And this is what it looks like when, if you want to see the video of these animals. So this is a spastic rat and a control rat. It is unable to move. You see, it's not really walking. And this is a spastic rat that has been treated with stem cells. It's hopping, it's running around. Motor function is very close to normal. The handlers who worked with these rats for decades cannot really tell the difference between a normal rat and a spastic rat that has been treated with stem cells. So that was a preclinical work that we did. We also had a phase 1 first-in-human clinical trial in Mexico where 7 patients were implanted with 7 Parkinsonian patients were implanted with these stem cells. Here you can see the gray bars are the baseline. This is the UPDRS scores, UPDRS Unified Parkinson's Disease score. Motor part of it is what they had before the surgery. PD is a progressive disease, so you expect them to deteriorate about 2.4 points per year. And the blue bar is what you would expect the score to be after 5 years. The green bar is actually where it ended up after these patients were treated. All patients are doing better at 5 years than would be an expected progression. And 6 out of 7 patients are doing better than 5 years prior. And this is an F-DOPA PET representation of it. In the middle, you see one of the patients. This is what Parkinsonian brain looks like. This should be much brighter, putamen. It indicates not enough dopamine production. On the right-hand side, this is still not a normal brain, but you can see it's much more dense. So the dopamine production increased quite a bit. I am pretty much out of time, but organizers, do I have a few more minutes? I'll just keep going. Just a few more minutes, just to show you how universal these cells are. We have a veterinary division as well, and we had a trial, a double-blind placebo-controlled trial where brain-derived fetal equine cells were tested to treat equine tendonitis. These are experimental lesions used with cholanginase in tendons. On the left-hand side, this is a treated horse. You can initially see the lesion. This is this big black hole. And then you can see it filling in, and by 8 weeks, it's almost gone. On the right-hand side, this is a control treated horse. You see the lesion, you see the lesion, you see the lesion, it doesn't go anywhere. And what's even more importantly, on top is a treated horse, and you can see this linearity. This is a parallel tendon arrangement. This is what you want to make sure that the horse does not re-injure. On the bottom is control, where you see the fibers are disorganized. This is not a normal tendon, this is scar. This horse will have forever weakened tendon. So stem cells are able to not only help with tendons, but they, they provide an anatomically correct reconstruction. In conclusion, what are the pros of using fetal-derived undifferentiated cells? It's a holistic approach to disease that allows you to treat the disease in its entirety. It also available for multiple indications because the cells adapt themselves to the disease. It can treat diseases with unknown etiology. We do not need to know what the mechanism of action is, such as with gene therapy, for example. We can put the cells in and let them figure it out. And they can actually be made for quite cheaply and in large amounts. What are the cons? Especially with fetal-derived cells, it's stigma, depending on which country you are. Related to that, it's difficult to acquire donor tissue, but we think it's very well worth overcoming those challenges through educating people and trying to make this work. Thank you very much. If you need to reach me in the future with any questions, this is our website. This is